Hello everyone and welcome back to Virtual Biolab. Today's video covers an interesting subject, which is cell culture. As you may know, for this area there is a variety of methodologies, different platforms, diverse materials, etc. that are each applicable depending on a few parameters, such as the purpose for which the cell culture is performed, the type of cells, the procedure itself, and other ones. But what is our method? Here's a list of materials that we utilize to culture human or other mammalian cells. For this we have FBS, which stands for fetal bovine serum, Pen-Strep, which is an antibiotic mixture, and the most important part, the culture medium, which is in this case the MEM. More information on each one of these will be given later in this video, and for now, let's dive into the lab and start the process. Here we need to take all steps for culturing under a laminar flow cabinet to prevent any possible contamination. This bottle here contains the MEM or the culture medium with a somewhat red color. And here in this 50 milliliter centrifuge tube we have the FBS. And the small tube next to it contains the antibiotics. Now we need to mix these three components in a way that the proportions in the final volume will be 50 to 20% FBS and 1% Pen-Strep. Before this step, Pen-Strep should be mixed thoroughly by pipetting repeatedly in order to resolve precipitations if there is any. Since we are preparing this mix in a 50 milliliter tube, we add half milliliter or 500 microliter Pen-Strep to 8 milliliter FBS, which is already in the tube. Then the medium is added to the tube until the mix reaches the final volume of 50 milliliter. But why did we mix these? The type of the MEM that we use here does not have any proteins, lipids, or growth factors, which all are necessary for the cells to grow. So to supply these and other nutrients, bovine serum is added. As for Pen-Strep, it is a mixture of penicillin G and streptomycin. These are antibiotics to prevent bacterial or fungal contamination of the mammalian culture media. The MEM itself stands for Dalbecko's modified Eagles medium and contains Glutamax, glucose, HEPs, sodium pyruvate, and sodium carbonate. Glutamax is a substitute for the amino acid glutamine, and unlike glutamine, Glutamax does not produce excessive ammonia in the medium. Glucose and sodium pyruvate provide the energy and the carbon source for the cells. And finally, HEPs, which stands for hydroxyethylpiperazine ethane sulfonic acid, together with sodium carbonate, act as buffering solutions to maintain the pH of the medium within a desired range. For the next step, we need to equally transfer the preparation to the flat-sided culture flasks. From here on out, I'm going to refer to this prepared mixture as the culture medium or medium alone, since it now plays the role. Care must be taken here to prevent any physical contact between the tip of the pipette and the opening or the inner surfaces of the flasks. Again, all these are for avoiding contamination. When the transfer is done, now we're ready to add the cells to the flasks containing the medium, which will be covered in the next video. So it means that that's it for today, and we thank you all for watching this video. If you found it helpful, please feel free to give it a like, and if you have any questions, I would be glad to know about them in the comment section. Also, your support means a lot to us, so please subscribe to the channel and hit the bell for more biology related content. And until the next video, take care.